Hello, welcome to the second lecture on plankton. So this links to your first lecture, which Chris Lowe, I think, gave you a few weeks ago. So I'm going to briefly review some of the stuff that he talked about, and then also talk about how you can sample and observe plankton and how the distributions change both in time and space. So in terms of the end of this lecture, we're going to have a brief review, as I said, about different types of plankton and groups. We're going to learn about the different methods used to study plankton and how they all complement each other. So the fact that no single approach is perfect, they all have advantages and disadvantages, and inherently you will find you have to use a combination of them for you to answer research questions. And then we end the lecture with how we can look and use these methods to actually study distributions of dynamics and how these may be changing through time. So first of all, just a quick revision from your first plankton lecture with Chris Lowe. This is one way of grouping different types of plankton. So we have phytoplankton, which are plants, zooplankton, which are the animals as consumers of some sort, bacteria-based plankton, which are predominantly involved in recycling, and also the funguses, also recyclers. So in terms of phytoplankton, we've got different types: diatoms, cyanobacteria, dinoflagellates, and coccolithophores. We're going to talk a bit about coccolithophores within the lecture. There's examples of dinoflagellates you can see under the microscope in the bottom right-hand corner of the slides. So where the flagellum means whip, hence they have a tail. And then top right-hand corner, you actually have an example of cyanobacteria, as seen in a freshwater system. So the plankton are not just found in marine systems, they're also in freshwater systems. So for example, this is a photo that I took from Argyle Reservoir. This is the stone you can see on the right hand side. This wall here is the dam at the end of the reservoir. This is a drinking water reservoir that feeds the Falmouth area and is a few minutes walk from the campus. But this green sort of surface distribution you can see around these two orange buoys are cyanobacteria. So these are plankton which are living in the reservoir, and you can see this is what they look like under a microscope, the image in the top left-hand corner. You can also detect and observe plankton from space, and the author, in fact, of this study is now with us, has just recently joined us at the university as a lecturer. But I include this mostly because it's a nice description of why we're interested in plankton. So plankton play a fundamental role in the biogeochemical cycling of the planet. And there's a bunch of different communities which I've just briefly revised from the previous lecture. And they're the primary source, the primary food source within the oceans. So they're the base of the food chain. And if you understand the spatial structure of these different communities, it can then help you understand different more complex processes such as biogeochemical cycling and also the importance of food webs. So clearly they're very important to be able to study, monitor and preserve in some sense. There's lots of different methods for studying plankton. So I'm going to talk about the ones listed here. So we're going to cover water sampling, trawling, water sampling in terms of collecting using Niskin bottles, which is something you'll, I imagine if you haven't already, you'll be gaining experience and seeing them actually used in the field when you go on your field trip to Plymouth Marine Laboratory later in your degree. There's also trawling methods, so using nets, and the continuous plankton recorder, again, which I imagine you'll have first-hand experience with later in your degree. And I'm also going to talk about remote sensing approaches. So this is observing from a distance, is what we mean by remote sensing. So we can do this using instruments which we lower into the water, but also you can do this from space. And I'm going to give you, as I go through, examples of how these are currently being used. Some of these methods may appear very simple and basic, but they are still very effective and still in use for research today. So the first one is plankton net. So this is very simply a large net which is lowered into the water and then pulled through the water. And at the very bottom here, you can see with a mouse cursor, at the very bottom here is they've labelled it here as a sampling bucket, but it's quite often just some sort of like a mug, that sort of kind of size, and it has a filter paper in it. And so the water is pulled through the net, and then whatever collects on your filter paper at the end, you can then take out and then put under the microscope to manually identify the different species and groups which appear in your net. You can have all different sizes. This is an example here of one actually being lowered from a large research vessel on a crane. Equally, you can see in the bottom right hand corner here a very small one being used in a lake, just with a pole. And this provides you with an integrated snapshot of the communities which are in the water. By integrated, I mean that it's a sum of all of the different communities through the volume of water which the net is towed. So, and that in that sense is a integrated or a summed snapshot of the communities. So the next example is using a Niskin bottle. So a Niskin bottle is a method of sampling a body of water. You can see various examples here. Essentially, it's a large plastic tube which is lowered into the water. You then lower it to the depth that you're interested in, and then if it's a manual version, you can see this brown copper weight here on the wire where my cursor is. You lower the whole bottle down to your depth of interest, and then you run this. It's called a messenger. You drop this weight down the cable, and it hits this white spring-loaded release here, and this snaps the top of the bottle, and the top and bottom of the bottle shut, and therefore captures the water within the tube. So you can see this is the version of the Niskin bottle with it preloaded before it snaps shut. So you can see here the two pieces, the top and bottom of the tube, and the tube is empty. And then after it's been fired and the weight has been dropped, the two ends have actually closed and captured the water. Again, there's lots of different versions of this. These are small handheld versions you can see here. And for example, there's someone lowering them off the side of looks like a fishing vessel. Where you can actually get much larger versions and rosettes. So this is a rosette. So there's a collection of lots of different Niskin bottles that are all being used from a research vessel. And you can see an example. These are techniques which are being used right now. So, for example, this is a research cruise which goes out annually. It sails from the UK down to Antarctica every year, monitoring the ocean and as a research cruise. And you can see here they have a rosette which is being lowered into the water from the crane of the ship. Okay, so back to the slide. So the idea is you collect the water of samples, you then fix it in a dilute acid which kills the plankton, and then that enables the water sample to be analysed at a later date to identification of the different groups and species within that water sample. Now this method is a snapshot in time vertically, so it's not integrated. Although I suppose you could consider it to be integrated over the length of the tube, but it's a snapshot vertically, so you lower it to your depth of interest and then collect a water sample. The advantage with using this rosette approach here is that it means you can actually collect snapshots at lots of different depths. So with a rosette, they would fire or trigger each of the bottles at different depths and therefore collect samples all the way down through the water column. So the next example is a thing called a continuous plankton recorder. So again, this is a towed sampling method. This was actually first invented in the 1930s by Sir Alister Hardy, and it is still used today. It's pulled through the water from a connecting line, which this is an old version here. You see this swivel. The connecting line here goes up to the ship. It's pulled through the water, and as the water passes through the inlet, so you can see on this diagram at the bottom, the water passes through this inlet, it then is pushed through a filter paper, a rotating filter paper, or mesh. So the mesh slowly rotates and coils up. So this is not an integrated measurement, but it's essentially you end up with a very long piece of mesh, and each small section of it corresponds to a different portion or different position within the ocean. These are still being used. These are used in the Atlantic predominantly, but they're also increasingly being used in the Southern Ocean and other parts of the world. And in fact, if you were to go on the ferry from Plymouth to Roscoff, the Brittany ferries, the Amérique, the ferry that operates there, actually tows one every single time it goes across the channel from the back of the boat. And this organisation, the SAFOS, is also based in Plymouth, and they're the organisation that runs these plankton recorders. Right, so the next kind of method for studying plankton distributions and species and groups is the approach of remote sensing. So by remote sensing I mean viewing from a distance. So we essentially remote sense when we look from our eyes, because we're observing the environment around us in some sense without necessarily touching it or interacting with it. So examples are our eyes, video cameras, weather satellites and thermal cameras. You can see that there's a whole range. This is the electromagnetic spectrum on the bottom here, and there's a whole range of different types of remote sensing approaches which will exploit different parts or portions of the electromagnetic spectrum. The light that we see through our eyes, so around 400 to 600 nanometers, is this visible light spectrum in the middle of the electromagnetic spectrum here. But for example, thermal infrared, so if you're sensing temperature, you'll be moving into the infrared radiation portion, and if you're using active remote sensing techniques, you'll be heading towards microwaves. So an example of in situ remote sensing is a method called flow cytometry. So this is one approach where you could take your water sample you've collected with your Niskin bottle, and then you can push it through a flow cytometer to actually study the distributions of the plankton within it. So but this operates, it's a pumped system, so you pump your water through. It's pumping the water through a very small space. This is a 1 millimeter cubed area of space, which is and the water is being pushed through at 1.6 meters per second in this case. So it's quite a fast flow for a very small space. So obviously this approach is good if you're looking for small plankton, but it's no good if you've got large plants or animals going through, because clearly it's going to block it up. It works by using lasers to excite the contents of the water sample, and they fluoresce, and as a result of their fluorescence response you can then identify which groups or classes of plankton are actually within your water sample. So this enables species and concentrations to be determined very quickly and very easily, and you can see someone here operating a flow cytometer. So these are relatively small pieces of equipment, and you can have them on ships, on research vessels, so you can immediately analyse your sample as soon as it's come from your Niskin bottle. Another approach of remote sensing is using Argo floats and bio-Argo floats. So this is an Argo float here. You can see this is one here. You can see on the right hand side it's quite tall. This is about, I would say, about 7 feet tall. They're quite tall instruments. And this is Giorgio. This is a colleague who works at Plymouth Marine Laboratory. And these are released from ships, so you can see this is what they look like out of the water, whereas on the left hand side you can see this is what it looks in the water. You can see the aerial, the transmission aerial coming out the top here, and the Argo float sat below. Now the Argo program in general is a multinational program. It's supported by over 30 different countries internationally, and they currently have over 3,800 floats all in the water delivering data. The majority of these are used to observe temperature, pressure and salinity. However, there are an increasing number now which have biology measurements being collected on them via fluorescence. So a similar method to the approach used with the flow cytometer, but in this case it's being used on a float. So I can give you an example of this. If you go to this link that I've given on on the lecture slide, you can actually see this is the BioArgo website. And you can see this is how they, as of the 6th of November this year, they have 3,855 floats operating in the water, and these are the locations of all of those floats. So that is a lot of floats. And you can even, they're on the same website, they have a link for looking at it in Google Earth. So for example, this is the view you can see in Google Earth. So if I zoom out, these are all the floats and their current positions as of the last time they logged, and I'll explain in a second how they logged that. So if we then zoom in, you can select a particular float and it will come up and tell you who paid for it. So in this case it's the United States. This is their it's a Southern Ocean float that they've funded. You can see when it was deployed. So this one was deployed in January, 29th of January 2018, and it last gave a location on the 15th of September this year. That doesn't mean it's broken, that means that was the last time it came to the surface. And you can see these are all the sensors it has fitted to that float. So it has a fluorometer, so it's looking at chlorophyll A. This is our biological remote sensing approach here. It has a spectrometer for looking at nitrate. It also is looking at dissolved oxygen, water pressure, so that means it can get depth. And it also has the standard suite of temperature. This is conductivity, so can be then converted to salinity and pressure. In addition, this particular float also has a pH sensor, which is quite rare. But you can see, that all of these floats are all currently in the ocean, collecting data and sending data back via satellite communications. And these are predominantly used for weather forecasting and ocean forecasting, but also used for understanding biogeochemical processes, the impacts of ocean currents, eddies, and so on, circulation patterns, and so on. So the web links for these are all within the lecture slides. So obviously this, 3,800 floats in the water. This creates a bit of a problem. Is what happens to the float when it stops working? They have internal batteries. So when they fail, sometimes they might fail at depth. Sometimes they fail at some surface. Sometimes they're collected. Sometimes they're not. So obviously this is an interesting point in terms of, especially at the moment, with the issue of marine litter being very clear in the media, and a clear push to reduce marine litter. There's the open question in the community: how much litter is actually produced from programs like this, from instrumentation, which essentially becomes lost at sea, and where does it go? The way the Argo floats work is that you release them from your ship. They then cycle down, so that you drop them from the ship. They then descend on a pre-programmed descend and rise process. So they descend down through the water column collecting data. They'll then drift, depending on how long you've set the drift for, at a predetermined depth, and then they'll come back up to the surface. And at this point, they then transmit the data via satellite back to a data repository of some sort. And all of these floats are continually doing this, so they continually come to the surface, they then drop down, and obviously as all this time they're being pushed around by the circulation, the ocean circulation. And here's a list of the different types of measurements which they can collect. This is just this is a snapshot from 2015. You can see that these are, at that time, these were all of the different floats, whereas the ones in green are the bioptical floats, and the ones in red were dissolved oxygen. Now that situation has changed a lot, and there's far more of the dissolved oxygen and bioptical floats, and certainly nitrate floats. An example, I didn't have to search very long until I found that one I just showed you on Google Earth, for example. So those are in situ methods of remote sensing of phytoplankton. You can also use the same kind of approach from space. So these are from satellite observations, where the satellite is orbiting at about 600 kilometers above the ocean surface. So predominantly for plankton, the kind of typical or standard data product you would get from a satellite observation is total chlorophyll A. And these are from ocean color observations, so these are from satellites which see in and observe the ocean in the same similar wavelengths to what we do. So they're working predominantly on visible light, so 400 to 600 nanometers. The reason why they work is that the principal absorber of light in the open ocean are phytoplankton, and the major pigment within these phytoplankton is chlorophyll A. So chlorophyll is a green, as I'm sure you know, green pigment produced during photosynthesis. So just as you have photosynthesis with plants on land, you also have them at sea. And so these ocean color sensors are used to estimate the chlorophyll A concentrations. So the sensors measure the amount of light that leaves the water, and from this they can use empirical algorithms to estimate the total chlorophyll A within the water and within the surface water. So obviously there's various different types of chlorophyll, from A, B, C1, C2 and D, but the main types we find in phytoplankton are A and B, but the majority of the algorithms will actually tend to focus on and concentrate on estimating chlorophyll A. So just to explain how they work, this is a plot of absorbance of light in the visible spectrum range. So here we have our 400, in this case up to 700 nanometers for our visible light. And then we have the reflectance spectra, or the absorption spectra, sorry, of the different types of chlorophyll. So this is, the red line is chlorophyll B, where the cursor is there, and the green line is chlorophyll A. And you can see they have similar shapes, but they're shifted slightly in wavelengths. Now, obviously you can tell different things from these absorption spectra. So the area I've ringed in red, this is where, if we're particularly interested in A, chlorophyll A, this response is explaining why plants appear green, because green light is being reflected in this area of the spectrum, because it has a low absorbance in that part of the spectrum. So these chlorophyll algorithms that the satellite observations exploit, they're typically based on empirical relationships. So they're developed based on collecting large amounts of in situ data from ships, from sampling stations, from buoys, and then the spectral response of different concentrations of chlorophyll are measured, and then empirically you derive an algorithm that relates the spectral bands that the sensor in space can actually observe versus, and the output of that is an estimate of the total amount of chlorophyll at the surface. And typically they result in these ratios of spectral bands, and they're linear algorithms. And it's all based on the analysis of the spectral slope versus chlorophyll concentration. So here we have an example. This is for a sensor called CWIS, which is an old US NASA sensor. They're interested in this particular algorithm of wavelengths of light of 443 nanometers, or center wavelengths, and 555. These were the two center wavelengths of this particular sensor, or two of them for this particular sensor. These are the in situ data, which were all collected in the Mediterranean during 2003 and 2004. And you can see the linear relationship. This is the ratio of the two water leaving radiances. So this is the light that's leaving the water at these two wavelengths at 443 and 555 nanometers. And here we have our estimate of chlorophyll as observed from the satellite. And you can see there's this linear relationship between the two. Sorry, the estimate of the in situ chlorophyll. And you can see the linear relationship between the in situ chlorophyll and the ratio of light from the satellite observing. So if you now do this, and then obviously your satellite is orbiting at 600 kilometers up, it's seeing globally. It won't be able to see through clouds, just as we can't, because it works on a similar spectral wavelengths, but it does mean that you can then start to get global distributions of chlorophyll A concentration. So this is an example from NASA data. Obviously you can see all the land masses. Here's the UK, and you can see the variations in concentration throughout the globe as seen from space. So you have lots of different characteristics here. We've got subtropical deserts, or in the gyres, where you have very little food, lots of light, but not very much food, and therefore you don't really get very many very high concentrations of plankton. Whereas you've got examples of very intense upwelling systems. So this is where wind-driven processes are pulling up water from depth, and as they pull up the water from depth, they also, with that water, bring nutrients, and these combined with light and warmer environment means you then get growths and high biological activity in these upwelling regions. And of course, as we know around the UK, this is a highly productive shelf region. There's lots of nutrients all coming off the land and in the coastal seas, combined with light, which means you get also get a high biological productivity and larger chlorophyll concentrations. If you then monitor from space through time, it means you can then actually understand how the distributions change spatially and temporally. So this is a sequence that's been generated from NASA data. You can see on land you're getting the greening of the land as the seasons change. So you can see the shifting of the green belt here in the sub-Saharan Africa. But you're also getting the same in the oceans. So you can see here, for example, this is the influence of the Amazon. You can see the Amazon plume here coming out into the Atlantic Ocean, and you can also see the shifting of the plankton distributions as you cycle through the seasons. So in the same way you're getting seasons on land, you can get seasons in the ocean, and this can all be observed from space. So you may have noticed that I've said so far that the satellite observations are looking at surface responses. So the satellite observations are able to estimate the surface concentrations of chlorophyll A. Now the definition of surface changes depending on what is in the water. So here we have the penetration of light in this cartoon for pure water. So this is the idea: is if there's no particulates in the water, it is pure water, then this is the light penetration you're likely to get. And they also have these definitions between the sunlight or euphotic zone, which is predominantly where the plankton distributions will exist, through down to twilight, and then the midnight zones where there's very little light, or no light in the midnight zones and very little light in the twilight zone. However, the penetration depth on the actual chlorophyll concentrations, what they're valid for, changes depending on what's in the water. So if you're in a very highly turbid area, coastal or shelf seas area, you may be observing the signal from only a few millimeters into the water column. Whereas if you're looking at our oligotrophic area, our gyre, our desert that I pointed out in the Pacific, then that signal that you're seeing may actually be coming from 20, 30 or even 60 meters into the water column. And that's because there's very little in the water, and therefore the light is penetrating much further. So you have to take this into account when you interpret your data, as the depth of your chlorophyll A concentrations may actually be from different depths, even though you're using the same technique. And the other point to make is that the satellite observations are, again, a snapshot in time. So they're a snapshot of the point where the satellite sensor actually flies over, and so they're a spatial distribution of the concentrations in a snapshot in time. So for satellite estimated chlorophyll A, the depth is dependent upon the turbidity of the water and hence what's in the water, the concentration. So it can be less than 1 meter, or it can be more than 20 meters, depending on what's in the water. So you can't use, this is an example of why you can't use satellite observation data in isolation for studying regions. You have to use a complementary, a group, lots of different types of approaches to study chlorophyll A and plankton distributions if you really are going to fully understand what's going on within a region. because they all had different advantages and disadvantages. So now we know a bit more about the depth. What can we actually see from this is our example I showed of the global distribution of plankton. So we now know that subtropical deserts, this region here, we're getting very low values, but we know that this water is very oligotrophic, it's very clear. Therefore this signal is coming from, may well be coming from the contribution of anything up from the surface up to 20, up to 60 meters depth, maybe. Whereas we know that these intense upwelling regions, there's going to be much more material in the water, many more particles, and therefore they're really surface, maybe 1 meter or less depths that these signals are coming from, and the same for these highly productive shelf areas where you have a lot of suspended particulates in the water. I'm just going to end on the different techniques by mentioning sediment coring. Sediment coring is another way to look at plankton distributions, and this is a way to look at plankton distributions after they've died. So you can look at historical plankton distributions. This is an example of a corer being used, I think, in a lake. And the idea is you just drive it down through the layers of the sediment, and you end up with a core like this. And from this you can determine the different layers and different years of deposition as plankton have formed at the surface, died, sunk, and then settled on the bottom. Not all of much of the plankton, depending on the type, will have decomposed, but you will find that some types that have some sort of calcareous substrate or structure, or ones which provide cysts, so effectively seeds for later, that if resuspended can actually then grow into new plankton distributions. So by analysing the layers within your core, you can actually look at past and historic distributions of plankton. Okay, so that's the end of part 1, and I'm just going to stop the video and then we'll start part 2.